So this is lecture 8, and in this lecture we're going to be talking about DNA, and then moving on to talk about the origin of life on Earth. The context of this here is, the last couple of lectures may have sounded more like a biology class than a physics and astronomy class. But the purpose of this is to really get a sense of how life works on the planet Earth, so then when we go off and start thinking about trying to find it on other planets, we might have at least some sense of what to look for. Another reason why I'm spending a little time today on DNA, and that's also why we talked about photosynthesis and the ATP-ADP cycle yesterday, or on Tuesday, is to really get your brain around how complex this stuff is, even on the sort of the scale of single cell organisms. So how amazingly complex the ATP cycle is, and you'll see here today that if you haven't thought much about how DNA works, it's also just an incredibly intricate thing that allows heredity to be passed on, and fundamentally that's what allows evolution to happen and take hold for the history of life on Earth. So in the first part of the lecture, we're going to be talking about DNA and RNA. So all life on Earth uses DNA to transmit instructions, to sort of are the agents of heredity in cells. What DNA is, it's a double helix polymer. So it's basically made of a sugar and a phosphate that's wrapped up in this thing that you might imagine looking like a ladder, but it sort of twists around. And fundamentally, there are 4 base pair molecules that encode this information. The genetic code, that is genes in the genome, are encoded in the DNA, and this is the mechanism for reproduction in cells. So when cells divide, this is the mechanism by which they divide, and they sort of carry on the same information as they split, and they continue like that. There's something else called RNA, which is similar to DNA, but the RNA is the thing that determines the cell's function. It allows the cell to synthesize new proteins and effectively tells the cell or carries out the instructions of the DNA, you can think. Now, importantly, as we'll talk about, when a cell divides, the DNA unwraps and splits apart. And because of the base pair configuration, it's sort of natural to build an exact copy of this DNA that then moves on to the next set of cells. But if the copying is not perfect, that creates what's called a mutation. And so if you change the DNA instructions in a cell, in principle, that allows for randomness in the process. That if there are external forces that favor whatever random mutation happened, those mutations will be sort of propagated through the next generation of life. Fundamentally, this is the agent of evolution. And the story of how people figured out that this is what's going on is what I'll start with in the beginning of this lecture. So just so we're all on the same page here, this is what we're talking about. So we have a cell, right? So life, you can break down in sort of individual cells. And in complicated cells, there's actually some subset of the cell called the nucleus. And at the heart of the nucleus lives the chromosome. The chromosomes are agents of heredity, but the chromosome is effectively made up of this DNA. And so this is the DNA thing we're going to be talking about. So we talked a bit about the sort of prehistory of this before when we were going through sort of the history of the biological revolution. But sometime in sort of this 1870s, because sort of technology had gotten to the point where people could begin to actually study the hearts of cells, people had realized that hereditary factors reside in the nuclei of cells. And so, for example, the Hertwig was doing these observations of sperm and egg fusion in sea urchins, and realized that it was really what was going on in the nuclei of the egg cell that sort of was the heart of heredity. A little bit later, people began to understand that inside the nuclei were these chromosomes. They're sort of these thread-like structures that look like this. And people were beginning to understand that these were actually the main things that were playing the role of heredity in these cells. And so then people said, okay, well, let's try to figure out what's going on with these chromosomes. Around 1910, people had then figured out for sure that these chromosomes were fundamentally the sites of heredity. This is where the genes were living, in these chromosomes. And if you remember the story about the fruit flies, Thomas Hunt Morgan was doing these experiments with fruit flies, and it was with these experiments that people really began to understand for sure that this is where the sites of heredity were. So if you moved on then into sort of the 1930s, most biologists at this point had begun to understand that there were molecules making up these chromosomes that were sort of the hereditary molecule. That is, that there was some kind of giant hereditary molecule that carried the information of heredity. And in fact, they knew what this thing was made of, and it was already basically DNA, deoxyribonucleic acid. They understood that that it was in there. But the fundamental thing is they didn't understand how it did the coding. Right? So if you just have a blob of molecules, how do you take a blob of molecules and then provide some kind of instructions for what to do? You know, how do you use that as sort of an instruction manual? So this was sort of the holy grail. Rosalind Franklin, pictured here, and she had a student named Maurice Wilkins. They were using a new technique that was called X-ray crystallography to get at the structure of DNAs. To get at the structure of DNA. And X-ray crystallography is something that you might learn about if you were taking a basic physics course. But they were harnessing this sort of power of X-ray crystallography to understand that DNA was a structured molecule. Now, so this is one of the image of Franklin's DNA studies. So a little bit later, after they were first doing this sort of pioneering work with X-ray crystallography, her student named Wilkins, showed James Watson the DNA image, like this, basically an image like this, without, his Ph.D. advisor's approval. So sort of showed it to the competitor and said, hey, check this out, isn't this cool? Okay. And later, James Watson and Francis Crick would basically use this technique to, the story goes, crack the code. And these couple of guys are generally credited with the first understanding of DNA. But fundamentally, they were using Rosalind Franklin's technique to sort of gain this information. In 1953, they showed that DNA can store and replicate information, and that it was constructed in this kind of double helix way. Effectively, what they did is they used the characteristic and features in the Franklin and Wilkins image to develop a chemical model of DNA that worked. And in 1962, they received the Nobel Prize for doing this. And so you'll often hear about Watson and Crick being celebrated for the discovery of DNA. It's an interesting sort of footnote to the story that Rosalind Franklin would almost certainly have been included in this prize, or she probably should have been included in this prize, but she had died 4 years earlier, before 1962. And when you get a Nobel Prize, you have to be alive. So they don't give Nobel Prizes to people who are dead. So this is probably why she doesn't get as much credit as she probably deserves in this story. But fundamentally, we have a picture now where people are beginning to understand the fundamental structure of DNA, and since that time, our understanding of that has now solidified. So what we know now is that DNA is this very long chain polymer. It's consisting of base pairs that I'll describe in a second. And via the sequence of base pairs, it does all these things. It does protein synthesis, it codes for RNA synthesis, etc. And the unit of sort of instruction is called a gene, and that codes for a single function. So just to give you a sense of this, human DNA, so our DNA, has about 3 billion base pairs in its code. We'll talk about what that means. And it's divided into 23 chromosome pairs. It's only 2 nanometers wide, but 5 centimeters long. And so effectively, if you scaled them up to the size of a ladder, it would be like a ladder that was 500 miles high. So they're these incredibly long string molecules that are twisted up that encode for all, a good fraction of what it is to be, to be human is encoded in this DNA. So the way it works is, and it's admittedly complicated, but it's doing a pretty complicated thing. You might argue that it's incredibly simple given how complicated the result is. So the DNA is this double helix, and so there's this backbone here colored in blue. And it has these DNA bases that connect the ladder, right? So these are like the rungs of the ladder, except there are different connections along the middle. So it's like the rungs of the ladder are split in two. And if you have like a blue rung, okay, or say a purple rung, you have to plug that into a yellow rung on the other side to keep your ladder together. So specifically these things that are called base pairs are thymine, adenine, guanine, and cytosine. And the important thing is that the adenine only pairs with the thymine. Okay? And the guanine only pairs with the cytosine. And so if you have a green thing here, you have to pair it up with a red. If you have a yellow, you have to pair it up with a purple, etc. And that's the only way it clicks together. And so that creates this sort of mapping that has to be preserved. And this is sort of the diagrams of what these different molecules look like. And they're effectively hydrogen bonds that lock them in place. When they get close enough together, they lock in. This is a little bit more detail about how these things work. Sort of give you some proportion on how big they are. They're a couple nanometers across. And the chemical structure is sort of given here in this picture. This is just sort of, if you're interested in this stuff, it just sort of shows you what it is. Now, how does this work? So how does the DNA, how do these base pairs code information? And why is it 4 base pairs? So why do you need 4 base pairs to code information? Why not just 2? Okay, so if you want to, for example, send Morse code to somebody, all you need is a dot and a dash, and in principle you can encode lots of information that way. Why 4? Yeah?
You can store more information in a smaller area.
As opposed to if it was just 2 base pairs? That's probably true. That's probably true. But why isn't it 35 base pairs then?
I think it should be.
Okay, so if you were designing life, you'd go 35 or some other number. Okay. That's interesting, right? So there is a pressure, I guess, then if you're interested in space, conserving space, you might argue that, oh, well, why aren't there just more of these? The more you have, the smaller you can squeeze it in together. It's a good point. So maybe if we find life on in another world, it would have more base pairs than this, or something like that. Yeah, good point. There's another way to think about it, which is there are 22 different amino acids that are used by life. So you can take a look at the amino acids that life uses to sort of build its structure, to make it go. And effectively what the DNA does is it wants to code for these amino acids. Okay? So imagine that there's a word for each amino acid. And each of these base pairs represents a letter in that word. Okay? A chain of words gets mapped to a sequence of amino acids to build proteins. That's basically how it works. So, for example, there could be 4, imagine the DNA coded information as single letter words. Okay, so imagine you just said every word in the English language was a single letter long. How many words could we have? So let me ask the question again. Imagine that every word in the English language was 1 letter long. How many words could we have in the English language then? Raise your hand. Yes? 26. Good. There are 26 letters. That means there's 26 words. That would be a pretty limited vocabulary, so that's probably not going to be good enough for us. This is a very different kind of alphabet because it only has 4 letters. So then, if you forced every word in this alphabet to be, every word in this language to be 1 character long, there would only be 4 things, only 4 different sets of instructions you could provide. But we're trying to code to build 22 amino acids. Okay, so there are 22 different amino acids used by life. And since there are only 4 words, that's not enough to explain how to make 22 different amino acids. Okay? So if I was trying to explain to you how to do something, and I only had 4 words, but there were options of 100 different things you could do, that would be a difficult situation. Okay. So now let's imagine that we had the same number of letters, so we still have these same 4 letters, but each word could be 2 letters long. That gives you a lot more opportunity here, right? Because you could have AA, AC, AT, AG, and then you can switch the first letter all the way through. And all in all, you could have 4 options for the first place, 4 different options for the second place. So you have 16 different options for 2-letter words with a 4-letter alphabet. But again, that's not enough because we've got 22 amino acids. That's still not enough. So let's say we wanted to say, well, you go to the next step. Maybe you imagine that you need 3-letter words. But if you have 3-letter words, then you're fine because then you have 64 combinations. That's plenty to make your 22 amino acids. And that's what DNA does. So what DNA does is it says every time there's a 3-letter word, that's a piece of information that says, okay, make this amino acid. There's another 3-letter word, make that amino acid. And depending on what the 3 letters are, you code for different amino acids. Okay. And so these happen to be all of the coding possible codes. Yeah?
Does it only read the left half?
In principle, it reads both halves and it sort of combines and then spits them off. And then you have sort of a mirror image over here. So one, sort of depends on what life is specifically doing to do those codes. So you mean the left half? You mean in terms of when the thing splits, or?
Well, because the problem is, you can only have certain AGs on one side and certain ones on the other side, so you can't just randomly match them up.
That's right. You can't just randomly match them up.
So I mean, codes won't fit always.
They would and then be a different sort of slice of code from the other side and this side. So it's a pretty complex scenario. If once you think about it this way, you realize it's a pretty complex scenario. That's right. So what effectively happens then when a cell divides, okay, so just sort of go back to sort of what happens when a cell divides, and this is sort of what you were talking about too. You've got your double helix, right? The helix unzips when a cell's getting ready to divide. Each single strand's matching bases are added by an enzyme. So there's some enzymes that come through and basically hunt for the matching base and build the other side. So you have your left side of your ladder, your right side of your ladder. When a cell's dividing, this thing unwinds. The enzymes build up they sort of build the complement to the left side of the ladder, so now it's a full ladder. They build a complement to the right side of the ladder. It's now a full ladder, and then the cell itself divides, and you've got a copy. But they can only be, the point is that in most cases, you get a perfect copy, because you have to. You can only add one base pair to this one, and so you get a perfect copy as the thing divides. So you're just replicating these sets of instructions during division. Right, so this is just sort of a diagram of this. You have these two strands that split apart. You build the other side because it's the only way you can build them based on the base pairs. The cells undergo mitosis, which is just they split apart, and then you have 2 cells that are now copies of the first. And this keeps going. So another thing that's interesting to think about is that there's another thing called RNA, which is a single-stranded helix that has a different backbone. And in fact, it uses uracil instead of thymine as a base pair. So it's a slightly different thing. And what the RNA does, okay, is it copies the instructions from the DNA. So the thing that's unfolded, it comes through and copies instructions, and then comes back out and carries out those instructions. It transports amino acid for synthesis, and it catalyzes protein synthesis. So RNA does these things, sort of acts on the DNA and carries out these instructions. So what happens then is you have these nucleic acids. They act as the basis for storage and transmission of heredity in cells. You've got your DNA that encodes the instructions, okay, for making proteins and RNA, and then the RNA determines what the cell's function is, and it manufactures these proteins and the enzymes that the cell uses. So you can think of the DNA as storing the operating instructions. It's like the instruction manual. And the RNA is the thing that carries out the instructions in the instruction manual. I'm going to sort of not go into the details on exactly how the transcription and translation works for there's two different types of RNAs. What I want to talk to you is about something a bit more basic. So the takeaway from this is nature has figured out a way to sort of encode instructions using this sort of ingenious sort of type of alphabet based on the base pairs. Because it's got this double-handed sort of double helix structure, it allows copies of itself to happen. It can copy itself again and again and again. So that's good. We need replication for life. But occasionally, the copies aren't perfect. Every now and then, one of those base pairs doesn't get matched up in quite the right way, and you get a copy that has an error in it. Okay? So imagine, for example, you've got a bunch of three-letter words, which is sort of what DNA is coding for, right? Remember, it's these three-letter words. And just as an example in English how we might do this, imagine you have a sentence, the big dog bit the red fox. And it's your job to copy that information. Okay, but you're not a thinking person who understands that this word wouldn't make any sense. You're some kind of robotic process, right? You're just a series of enzyme reactions or something that's doing this copying. So you could do something where, like, you read the B incorrectly and think it's a Q. So you can be the big dog kicked the red fox. And then you have a sentence that doesn't make any sense. However, maybe from context, this wouldn't be a complete loss, and you could sort of figure out what's going on. Another possibility is you accidentally insert a letter. But because you, by default, always read this as three-letter words, this really screws up the whole chain beyond that point. Okay? So now you would read the big dog, and if you just stuck an R in between the O and the D, it would be the big dog drove, gibbet, flip, and then it just becomes nonsense. Or you could take a letter out and it does the same thing. It ruins the whole rest of the strand. Or you could do something even more extreme. You just insert a word somewhere in there. And then in principle, that's not quite as catastrophic, but it still kind of screws up the meaning. So these kind of problems happen in cell replication. And a lot of times, it either is catastrophic and just destroys the function entirely and the cell dies. Sometimes it doesn't change anything at all and the cell just keeps, things keep going right as they were before. But sometimes these errors that are called mutations change things, all right? Sometimes there's no effect, but sometimes you can make a subtle change in the organism, like, for example, you could change eye and hair color by some kind of weird mutation. Okay, so people with blue eyes, somewhere along the line, there was some weird mutation that happened and blue eyes happened, for example. Another thing that can happen, though, is you could have bigger changes that are more harmful, that could cause diseases, etc. So, for example, here's a normal fly. It's had a specific Hox gene mutation, and it created a leg instead where an antenna was supposed to grow. Right? So these kind of things happen. And again, oftentimes when these kind of weird mutations happen, it's not so good. And this fly is probably not going to live very long. And probably not going to replicate, right? However, every now and again, you get a mistake that makes the whole thing a bit more interesting, right? So before we had the big dog bit the red fox, so you don't really care. But then suddenly you're like, oh, the big dog bit the Red Sox. Like, that's a much more interesting sentence, right? So you could have these kinds of things that fundamentally change the meaning and might provide some kind of advantage. Or you could do a base deletion that still, means something but might not mean what you thought it meant before. So these mutations in principle then can cause genetic variations in a species. And if it happens enough, Every now and then you're going to get sort of lucky and get some kind of mutation that's advantageous. Okay? So if a mutated cell survives, then the mutation can then pass on to later generations. If that mutation provides some kind of adaptive advantage, there will be strong selection for that mutation. Okay? So for example, there's a mutation associated with sickle cell anemia. Depending on what the conditions are, okay, if you've got if you inherit the mutation for sickle cell anemia, if you happen to live in a place with a lot of mosquitoes, okay, then you are more resistant to malaria, and you might be more likely to live and thrive in that environment. And in fact, it's true. People who are from areas that have lots of mosquitoes are more likely to have inherited the gene for sickle cell anemia. So while it's not perfect to have sickle cell anemia, it's way better than to have malaria and to die of malaria. Okay? And so if all the other people you're living around, if some fraction of them don't have that mutation, they're going to die very young of malaria, and you're not, and you'll live and you'll have kids and that kind of thing. There's another example of a mutation as a specific name in humans that occurs in genes associated with T cells, and it turns out that it provides HIV resistance. And so people with this specific mutation have delayed onset of AIDS, and they live longer because they have this specific mutation. So you see these kinds of things happening in sort of like in real time, right? So AIDS was not a disease that existed, really 100 years ago, right? But because this random mutation has happened, there's some selection for that. Okay. So this mutation fundamentally is the basis for molecular evolution. That's the important point. Okay. So before I move on to the sort of next part, which is basically the origin of life on Earth, are there any questions about that sort of kind of quick overview of DNA, RNA, and DNA as sort of the basis of molecular evolution? Now you could imagine now, and next week we're going to start moving beyond the confines of the Earth to other objects in our solar system, and then eventually we'll move on to think about the sort of vast array of stuff that could exist orbiting around other stars. You can ask yourself things like where we were getting before, does it have to be 4 base pairs? Okay? Could you imagine a situation where life could have arisen with just 2 base pairs? Or maybe 5 base pairs, or 10 base pairs? You could imagine some kind of chemical structure that was different, but performed the same qualitative kind of behavior, that had the same qualitative kind of behavior. And so it might be, for example, all life on Earth is based on this, has DNA as the backbone. If we were ever to discover something that sort of had the behavior of life but didn't have DNA, that would be a true indication that we've discovered life that's foreign, that's alien. Same thing about the ATP cycle for the cellular battery. If we were to find life somewhere that didn't rely on the ATP cycle, maybe that would be a hint that this was alien life. Okay. So a requirement for life is you have to have a means of storing and transmitting functional instructions, heredity. The implications for life elsewhere is, does life on other worlds have analogs of DNA and RNA? Does it have DNA and RNA? That would be really weird if it had exactly DNA and RNA, but does it have to have some kind of internal analogs that serve the same functions of DNA and RNA? Probably. It seems like you would have to have something that would sort of have the functional instructions of heredity, so that then the forces of evolution could then take over and drive things towards more complex forms of life. Otherwise, it would probably be stalled in some kind of proto-life kind of state where you didn't have any kind of heredity to drive you towards more complexity. Are there other molecules besides DNA and RNA that could just as well serve the same function? Do these molecules have to be carbon-based like DNA and RNA are? Could the words be longer, like your 34 word, you want 34 letter words? You argue that makes the most sense. So when you discover life, maybe it'll be 34-letter base pair words. So then, it brings us to a question of, well, how did all this get started, right? So all life on Earth uses DNA and RNA. All life on Earth uses ATP, ADP cycle to charge batteries. All life on Earth relies on this subset of amino acids. How did all this get going? Okay, and so that's the point of this second half of the lecture. So there are basically 4 major divisions in geological time that we talk about, going back to sort of the very beginning of the Earth around 4.5 billion years ago. The first epoch is called the Hadean epoch, and Hadean is basically supposed to be associated with before life. Okay? During the Hadean time, the Earth was very hot. It was probably molten for most of the time. There was a period where lots of comets and stuff like that, and meteors and big rocks were slamming into the Earth. Sometime in there, the moon formed. I'll talk about this in a bit. A giant Mars-sized thing came and slapped into the early Earth. A piece of the Earth broke off, and that became the moon. So the Earth was undergoing this kind of crazy catastrophic period during this early time, during this Hadean period, and as far as we know, there really wasn't any life. But after this time, things kind of settled down. All of the shrapnel in the proto-solar system kind of died down because it merged away and bombarded other stuff away. And things kind of settled down on Earth. And this is when the first life emerged. Okay, sometime around maybe as early as 3.8 billion years ago, life seems to have emerged on Earth. And again, this is kind of amazing because it was sort of just at the point when things were cooling down enough that you could have real liquid water. So as soon as the conditions were marginally possible, marginally conducive for life, life seems to have sprung forth on the Earth. However, it took, a very long time before the first multicelled life to happen. And we talked about this a little bit last time when we talked about the evolution of oxygen content in the atmosphere. It wasn't until about 2.5 billion years ago before the first multicellular life existed. So all life on Earth before this time, for more than 1 billion years, seems to have been single-cell life. And it was only in the last 550 million years or so that the first animal life arose. So animals are things that move around and eat. So how did you get this started? And we think, okay, so we have this picture where you have the first life. Because of these genetic mutations and sort of evolutionary forces, eventually you have this pressure to move towards eventually a more complex life. I mean, it's not necessarily a march towards complexity, but you could see an avenue for sort of gaining advantage if you were a more complex life form than a simpler life form. But how did this very first life emerge? A clue to this comes with sort of, in biology these days, biologists actually break up the tree of life into sort of 3 large groups based on sort of the microstructure of life and the nature of the DNA, et cetera, that makes up those life forms. The 3 big ones are eukarya. Okay, so eukarya are things that have basically more than one cell. And that includes us. Okay, notice this tree. This is sort of a tree that's got all these lines coming out of it. All right. This line right here is all of animals, up in this very corner of eukarya. Okay, so we think when we think of life, a lot of us just sort of naturally think of animals. But the way biologists understand the tree of life now is that that's not really the right way of picturing it. That life has all of this diversity, and animals are just a pretty recent small little tree in this tree of life, small little twig from this tree of life. There's archaea then and bacteria. Archaea are, a subset of archaea are these extremophiles that can live in very high temperature environments, but they are single cell. And then there's another whole class of single cell organisms that are called bacteria that you've probably heard of. This one gives some approximate dates for when we think these different classes of animal life broke off from this primordial tree, but I don't really care about those dates too much. A simplified picture of this, which I think basically tells you what you need to know, is there's this understanding that there's a common ancestor, and at some point that common ancestor split off between archaea and bacteria. And then at some point out of the archaea came the tree that gave rise to eukaryotes. So that's the things that eventually became animals. Now, prokaryotes, that's these two things. See this label right here? That just means a single cell organism, okay, that lacks a membrane that contains the nucleus. So you just kind of have a bag with stuff in it, as opposed to the eukaryotes, which are cells, but then within those cells are other little bags that contain, for example, the nucleus, where the chromosomes are, and that kind of thing. Okay? So the prokaryotes, so both of these two huge branches of the tree of life, are all single-cell organisms, and they don't have a membrane-bound nucleus, they don't have mitochondria, they don't have any kind of membrane-bound organelles inside. They're just this sort of very simple structure. And so one of the questions that people have is, you imagine that the first life that arose was very, very simple, okay? Probably even simpler than the simplest prokaryotes that we have around today. How do you go from that and then eventually into things that have these complex cells and eventually multi-cells, that kind of thing? That's one of the big questions. Okay. So what's the difference between archaea and bacteria? Okay, both archaea and bacteria are unicellular. They have no nucleus, okay? Their DNA is different, though, fundamentally different. So the DNA in the archaea contains both exons. So those are part of the DNA that actually code for proteins that we spent the time talking about. But also other types of DNA that's known as junk DNA. We're not sure what this DNA actually does. It's not obvious what it's coding for. This is a topic of sort of a lot of research today in biology. So the archaea has this extra junk DNA just like eukarya do, just like we do. But the bacteria don't. And so that's one of the big dividing lines between these two. The thing to keep in mind is people used to think that these two things were very similar. Now there's an understanding the archaea are fundamentally different things than the bacteria. As I mentioned before, we do not expect the first life on Earth to be as complex as present-day bacteria. Okay, present-day bacteria do all kinds of things, all kinds of incredibly complex things, even though they're sort of just, we think of bacteria as so simple, right? But they do amazing things, right? They have DNA, they code heredity. They have this ATP cycle, which is a battery to provide energy to cell function. And they have RNA-mediated protein synthesis, okay? All this stuff is a product of billions of years of evolution to happen, even in the simplest little bacteria. So we don't think that this sort of emerged out of nothing. This didn't just spring forth from nothing on the early Earth. How did this happen? Okay. There's one interesting idea. It doesn't get to the very origin, but there's this interesting idea between how you get from how you get to eukarya. How do you get to cells that have this sort of complex nuclei inside of them? Okay, and one of the ideas is basically there were these two branches. Okay, there was archaea, this one kind of class of sort of single-celled life. And then there's bacteria, for example, the ancestor of the chloroplast, the thing that gives rise to photosynthetic energy generation. Or the ancestor of mitochondria, which is something that we know exists inside, for example, our cells. And we think what may have happened is these single archaea cells started eating a bacteria cell, for example, and ended up digesting it or anything. It just became sort of a subset of it that sort of ate what would eventually become its nucleus in some kind of simple sense. And so there are these ideas that effectively you have sort of crossing of these two branches created the eukarya, which gave rise to then the complexity of animals and plants today. And this is just a detailed picture of this. So that's sort of the type of ideas that sort of are backing back. Okay, so how do you get to sort of the eukarya? How do you get to these complex cells, starting from these sort of very simplified single-celled organisms that we think things must have started with? But what about before that? So how do you even get your first very simple types of cells. Where does that come from? Well, you can think about this in terms of, well, we've got raw materials on the early Earth. So you're back 4 billion years ago. You're looking around on the surface of the Earth. What do you have to play with to start imagining life springing forth? Well, we've got some raw materials. So eventually we had water. Okay, we had liquid water. We have some carbon dioxide. We've got a lot of organic molecules. That means organic molecules that have carbon in them. So we have sources of carbon. And you've got phosphates. So there's sort of these basic ingredients that if you just sort of break down what life is made of, it's basically made of this stuff. So you have the raw materials. So you have the things that sort of can make chemistry happen. Okay, and you've got energy sources. Okay, so you've got sunlight. Okay, that's an energy source in principle that can be harnessed. You've got heat because you've got a planet that's cooling off. And you've got inorganic oxidation. So you've got ways of sort of harnessing energy that way. One thing I just want to remind you of, the early Earth is hot. There's probably significant volcanism, including at the bottom of the oceans. There were almost certainly sort of undersea sources where you could have sort of heat vents at the bottom of the oceans. And this is one place where you have an energy source, you've got abundant water, and you've got organics. And that's one of the places that people are thinking now might have been sort of the key place for the origin of life. The other thing you're going to need eventually, if you want to have something that's alive, is you're going to want to have some kind of boundary. Okay? The cell wall. You need something that separates you as a life form from the background stuff. Okay, so how do you get this boundary, and where did the cell linings come from? You need some kind of mechanism for energy utilization. So you've got an energy source, but how do you trap it? How do you take that energy and then do something with it? So how did that happen? So people talk about whether or not you had metabolism before anything else. And of course, you need a mechanism for reproduction and eventually heredity, because heredity is this thing that allows you to harness the sort of general power of evolution to build up more interesting things, an adaptive type of life that could then really be successful. So some hints from this can just come from studying things. You just sort of look back on the history of the Earth and try to get a sense of when the first life arose. And we have pretty good evidence that some interesting stuff was going on maybe around 3.5 billion years ago, perhaps even earlier. How do we know this? So very quickly. We've got fossil remains of layered bacterial mats that people have identified that are possibly as old as 3.5 billion years. And we also have microfossils, fossils themselves, of little individual single-celled organisms that are dated also close to 3.5 billion years old. Okay, so these single-celled organisms seem to have been sort of percolating, within 1 billion years of the Earth's formation and not too long after the point where it got cooled down. There's another thing that I'll talk about next, which is carbon isotope ratios. This is a much, it isn't as direct evidence as this, but it turns out that life tends to take up certain types of carbon isotope, a certain carbon isotope more than sort of general non-biotic chemical reactions. And so by measuring these isotope ratios, you can get a sense of whether this came from organic matter, whether this came from stuff that was alive. And these are dated as old as 3.85 billion years ago. So, for example, there are some very old sedimentary rocks that seem to have a signature of this carbon isotope issue. So it turns out that plants preferentially incorporate carbon-12 over carbon-13. So by looking at the isotopic ratios of carbon in these ancient sedimentary rocks, people have found this enhancement that very well might be evidence that there was some living stuff that was existing some 3.9 billion years ago. So this is where this evidence comes from. It's not super direct, it's very different than sort of digging in the ground and finding a fossil skeleton of a dinosaur. Okay, this is much more kind of subtle stuff. But there are lots of lines of evidence that suggest that something interesting was going on at these early times. Okay. These first life forms were these prokaryotes. Okay, so there were these single-celled things. They didn't have nuclei, like I was saying before. And importantly, as we talked about last time, they did not require oxygen to live. Okay, they were anaerobic, they were not requiring oxygen. And almost certainly, any oxygen that they encountered would have acted like poison to them. So they were living in an environment that was oxygen poor, and eventually, you know, their descendants, eventually, right, the descendants of first life would be these cyanobacteria that eventually started producing oxygen. But the very first things that arose were not requiring oxygen for life. So they were very different than sort of the oxygen requiring life that we're familiar with. One of the leading ideas is that life actually began in deep ocean vents. We see these deep ocean vents today. They're in areas where you might have sort of a continental rift at the bottom of the ocean. So you have heat sources, for example. And so they're hot, they're rich in nutrients, they've got magma, so they've got all of these kind of interesting organics, right? And we know that microbes today can process energy through chemosynthesis. That is, there's no sunlight energy available at the bottom of the ocean. And what they use is they extract nutrients and energy from chemicals to keep them going. And we see this today. And so, there, you know, these types of archaea, this water can be extremely hot. So there are types of archaea that you see thriving in these kind of vents. This is one example of where life may have gotten its first foothold. For example, there are these class of archaea bacteria that thrive in temperatures above 80 degrees Celsius. So this is hot, okay? This is almost boiling water. They're more in water that's more acidic than battery acid, okay? They don't require oxygen. In fact, if they encounter oxygen, they die. And they get source of nutrients and energy from basically volcanoes. So if you go back in time, you know, 4 billion years ago, that's kind of what the Earth was like. And so this is not a crazy thing to think of as sort of a, you know, an analog to some of the earliest life on Earth. There's also these things called methogens that are anaerobic, that is, they're kind of oxygen toxic. They use carbon dioxide to produce methane, okay? And you see them in sewage and swamps and volcanic vents. So there's this type of bacteria, basically this sort of archaea bacteria that produce methane as part of their life cycle. And so it's stuff like this that we think may have been analogs to what was going on in the early Earth. One of the really kind of breakthrough experiments that got people very excited about the origin of life is called the Miller-Urey experiment in 1952. What they did is they showed that amino acids, these fundamental amino acids that are sort of the backbone of life on Earth, they could be made in the laboratory under conditions that we think are not unlike the early Earth. Okay, so what they did is they've got a mixture of water and methane and ammonia and CO, and they pumped it in this atmosphere, and then they ran an electric shock through it. So you can imagine this was lightning on the early Earth, okay? And then they precipitated water as rain, okay? And then they collected this sort of rain, the stuff that rained out, into, you know, sort of an ocean. So you can imagine the early Earth, you've got all this stuff going on, you've got lightning, you've got heat sources. So within this model, you put all this stuff together, you run some energy through it, and some water rains out into some early ocean. This early ocean is now rich with organic molecules that have been created just from these very basic materials, okay? These are not, you know, these basic molecules here, methane, water, you know, hydrogen molecules, CO, these molecules right here, we see them all over the place. They exist out in space, okay? You see them. You don't need a planet. You don't need to be on Earth to have this kind of stuff, okay? So that's the point. You take this stuff that just forms naturally. You put it together, you sort of add an electric shock to it, and out precipitates these crazy complex amino acids, okay? So they found that in this, you know, murky ocean water that they created, there is a soup of amino acids lurking in there, which are then sort of the fundamental ingredients for life. Now the problem is this experiment, turns out, had some problems. Okay, it got people really excited, but it wasn't quite perfect in the end. Okay, so one of the things is that methane and ammonia, things that they stuck in, we actually don't think that they were common in the primordial atmosphere now. So they had stuff in there that we don't think was probably appropriate for an early atmosphere on Earth. The other thing is that they produced only 5 amino acids, and there are 22 amino acids that take part in life on Earth. Okay. The other thing that they did, which is sort of a left sort of question, is they produced left-handed and right-handed amino acids. If you remember, all life on Earth is almost all is using only left-handed amino acids. So it's, there are still some mysteries that are lurking there. You're not creating the full sort of deck of cards that you need, that we see here with life. Interestingly enough, they did a second experiment that didn't get a lot of press, and a recent reanalysis of those data have been probably even more interesting than their first experiment. What they did is they actually, they basically used gases that were volcanic in their initial pot, so in their initial proto-Earth, okay? And we think the proto-Earth was very volcanic. So if you just go out and sample what volcanic, outputs of volcanic gases are today, you see that it's basically made of this stuff. If you do an experiment like they did before, but now sampling the atmosphere this way, you make all 22 amino acids. So when you look at something that's probably even more realistic of conditions of the early Earth, it's pretty easy to generate all 22 amino acids. So this, again, is kind of interesting. It suggests that at least getting from very simple molecules, the kind that you see abundantly all throughout the universe, how to go from these simple molecules to the complex amino acids is something that you can do. It's not like it's prohibitive. It didn't take any magic to give us these amino acids. Now, of course, going from amino acids to life, that's a different thing. Okay. Another thing to keep in mind is these complex organics and amino acids actually exist. We see them out in space too. So for example, this is a meteorite, okay? And if you look at isotope ratios in meteorites that clearly formed outside the Earth and they crash into the Earth, we see that there's all kinds of organic compounds out there in the solar system. So there's a rich, I guess, reservoir of these complex organic molecules that were probably zooming around the early solar system that could have been bombarding the Earth. And also, it's not that hard, even given very basic conditions of what life on Earth, what conditions on Earth were like, that you could have generated these amino acids. Okay? So that's kind of interesting. So that gets you to the amino acids. But all of this is still called prebiotic chemistry. Okay? Making amino acids is not the same as life, right? Amino acids are just molecules. It's chemistry, it's complex chemistry, they're big, crazy molecules, but that's not biology, okay? So the transition between chemistry and biology is like the big question. How do you go from chemistry to biology? What is it? What is that difference, and how did that happen, right? So this is a fundamental question. We don't know the answer to it, but it's sort of one of the deepest questions at sort of the interface of biology and chemistry, and arguably in all of science. How did life begin? Okay. So one thing you can think about is like, well, okay, well, where did the cell membrane come from? How does that happen? And it's actually interesting. This actually is something that probably happens pretty naturally. Okay. So there are things called lipid vesicles, okay? These are sort of like cell membranes. Effectively what they are is they're sort of like soap bubbles. They're like very similar to soap bubbles. And if they grow large enough, these kind of things, they can actually break into two. So you can imagine these sort of early sort of soap bubble type things, these lipid type sort of spherical blobs that can exist. You can imagine these things forming, and again, that's not life, that's chemistry, but you can imagine some situation where you get some sort of container, that interesting chemistry starts to happen in the middle of that container, and that separates you from the external world, and that's sort of the backbone of these, of some proto-cell kind of thing that could exist, all right? So you can think of these as containers for proto-life, or perhaps something like this. of course, you have the raw materials, right? You've got amino acids, say, and you've got these containers, which are not that hard to, in principle, create. But again, that's still not life. You can't just put RNA in a soap bubble and it's, that's not life. Okay. Sorry, I slipped up. You can't just put amino acids in a soap bubble and that's life. Okay. There are basically two scenarios that people talk about of how you go from this early chemical world to the biology world and how that happens. And one is via an intermediate step that's called RNA world. So the first forms of life or pre-life were based on RNA rather than DNA. So abiotic RNA, okay, living perhaps within a membrane, would be sort of a precursor for life. So that's one idea. And the other is a metabolism first idea, where you have catalytic networks, so ways to process energy in some way that's sort of a very, very simple form of the ADP-ATP cycle, for example, that could sort of process energy, harness that energy, and this idea is sort of you have a metabolism first. So you have metabolism before you have heredity. So now you have an energy source, and then eventually that energy source gives rise to some heredity. So these are the two ideas that people talk about. An RNA world hypothesis is basically the idea that there was RNA-based life that came first, before DNA-based life. This is an idea that's come about because RNA, for example, is simpler than DNA. Okay? RNA also stores information. So in principle, you could have simple heredity with just RNA. Okay? It catalyzes its own replication. And so you can imagine that you have something that's just an RNA strand that sort of acts in some way like a very simple form of life. The problem is, how do you get the RNA initially? How do you make RNA prebiotically without life to start with? Okay, you could start with basically the basic ingredients in some water with phosphates. So you could imagine eventually there's some chain reaction and you get complex molecule just kind of emerges. In fact, people talk about clays providing a substrate that could then help the nucleotides click up and form into these chains. So you could imagine that you had, if you were to create these RNA strands, a free-floating RNA strand could find its way into one of these lipid cells, just sort of these bags. Then the RNA is now protected, okay, and it can self-duplicate from other nucleotides that might be floating around in here. And so, if at some point this thing splits apart, you would have some early version of replication. So these are the ideas that people are throwing around. And then, the idea is once this happens, then eventually you find your way into sort of two of these things coming together, and it begins acting like a DNA molecule. This is all very nebulous, right? So this is how sort of unclear this picture is. So people have these ideas, well, in principle, RNA is simpler, so maybe that came first. Then you can imagine finding its way into some kind of cell wall, and then eventually, via just sort of kinetic energy, it starts dividing. It's a little bit unsatisfactory, though, right? Because you don't have any kind of there's no oomph there. It's sort of a slow process. But anyway, these are the idea that you have this sort of RNA world initially, and eventually, okay, you have the emergence of DNA, and then once that you have both DNA and RNA, these two things start acting together to provide sort of the DNA world that we see today. The other idea is you have metabolism first. And so this is the point that you figure out how to harness energy from your environment first before you have any kind of heredity going on. This is nice because you have an energy source. In principle, if you have energy first, if you have metabolism first, once you have this energy source, you could then have a way to assemble perhaps these complex molecules like RNA. So it sort of it answers this idea that look, you're not just going to, it seems crazy to imagine that you would just spontaneously produce RNA. It's a big, long, complicated molecule. Why would this just happen? Now, so the alternative then was you start with some kind of chemical network that processes carbon dioxide that we think was rich in the atmosphere, and uses this cycle to basically harness energy. And if you put these little sort of battery packs inside these same kind of cell walls, you would have these little battery packs then that you could then use that would then eventually provide an energy source for building more complexity along the way. Okay. So you have this general possibility then as you have these sort of boundaries of this lipid boundary. You have an energy source of some kind. For example, you're using inorganic minerals as your energy source. You couple the energy release to some kind of driver reaction that then, within this network that sort of keeps reproducing itself. Okay. And then eventually when the soap bubble splits, you've got sort of two versions of this that sort of keep going on. The problem with this is you've got no heredity. So you've got no reason to sort of build something more complex. You just have this sort of like internal gizmo that's not doing anything. It's just sort of, you have an energy source to sort of keep having an energy source, but you're not sort of driving something towards heredity that's building new proteins, et cetera. But these are ideas that exist, and it could be that there's both of these sort of forces were operating at the same time. So you sort of have a commingling of these ideas that eventually gave rise to something more complex at early time. The amazing thing about this, though, right, is this happened whatever gave rise to life on Earth, it happened very quickly. And so these processes, they have to be, you would think they would have to be pretty natural. It's not you would naively think that it's not some crazy random set of events that had to happen. Unless, we're the only life in the universe. Then you could imagine, look, there's 100 million planets in the galaxy, and 1 in 100 million, you just get this crazy set of random chemical reactions that gives rise to your initial simplest form of life, and then the forces of evolution take over. So that's one possibility. Okay. There's another idea that kind of circumvents the problem that says, the reason why life got going so early here is actually it was brought to the Earth by some mechanism. Okay, so you can imagine if there was some kind of archaea, some kind of extremophile life that was sort of living, or maybe in some kind of hibernative state on the outskirts of a comet or a meteorite that slammed into the proto-Earth, eventually this life could then procreate once it got this sort of place to take foothold on Earth. Okay. Now, this is an interesting idea, by the way, because there are extremophiles on Earth that you can basically put up into space for a long time and they don't die. So there are things on Earth right now that you can envision just sticking on a meteorite and sending that putting it on a rock and sending that rock to Mars, that would probably make it to Mars. Right? So you could engineer such a scenario. Of course, this doesn't actually answer the problem, because the real question is, how do you get life going from inorganic? Sorry, how do you get life going from chemistry? How do you start with chemistry, which is sort of what we understand? How do you make life from that? If you just say, well, life on Earth came from outside the Earth, then you still have the question, okay, well then how did life get going on that thing? So you still eventually want to answer these problems, but the initial conditions could have been a lot different than what we've got here on Earth. Does anyone have any questions about this? Okay. So that's the end, sorry. So this basically ends our sort of foray into sort of biology. Okay, so the basis of what biology is on the Earth. It's been incredibly coarse and simplified, and at some level there's some details there that I'm not going to test you over, right? So don't get freaked out about the details. I'm not going to ask you what the nucleotides are and stuff like that. But I did want to go into a little bit of the details, because I don't want you to think that it was simple, right? It's incredibly not simple. And somehow, amazingly, on Earth, this stuff got going pretty fast. And after 1 billion years of evolution, you get to the point where you have the simplest kind of cellular life that we see today. So most people who study astrobiology are reasonably confident that life like that probably exists elsewhere. Just because it got going so fast on Earth, people think that some kind of simple life must exist beyond. But then it took, 4 billion years to get anything that was, like an animal. And it took basically the entire history of the Earth before something existed on our planet that could send radio signals and receive radio signals. And so the next steps will be, we're going to explore the solar system, explore our solar system, within which we don't think that there are anything else sending radio signals. That would be very unexpected. But the real question is there microbial life out there? Because if there is, then it would sort of say, look, microbial life somehow got going on Earth, it got going on Enceladus, it got going on early Mars, whatever the answer is. If we saw that it was common throughout our solar system, then it would be very hard to believe that that kind of scenario wasn't common throughout the galaxy. Now you've got 50 billion or so opportunities. In each of those 50 billion opportunities has billions of years to eventually progress. And so then you can realistically ask the question, are there other things out there that are capable of sending and receiving radio signals? And that leads to sort of the question of intelligent life. So that'll be the rest of the course, is getting to that point. Okay, thanks.